Okay, so well, first of all, I would like to apologize. I was not able to be here the first 2 days of the meeting, but yesterday I had a full update from Stan Murray, which was here, telling me in all details every talk. So I was very excited, and that allowed me also in the train to take away lots of slides because a lot of the explanation of things like cell polarity have been given before by Alan, for example. So I really want to focus on some of the research we're doing in the lab. We're combining ideas of modeling cell polarity to the tissue level, but we're also trying to approach this experimentally. So it's a little bit of an adventure, and there'll be some detours in the talk about some things that we had to solve from the experimental side, but through mathematics. So what we're really interested in focusing on that system that I just showed you of the leaf epidermis are these very strangely shaped cells. I don't know if you're all familiar with them, but these are called pavement cells, and they look like little jigsaw puzzle shaped cells. So they have very complex cell polarities, which you can see through the cell shape. Now what's very interesting, associated to these lobes and indentations, we have what you guys have already been discussing, small g proteins. So we have certain proteins which are specialized to give these regions of the cell the ability to protrude and to indent. Okay, so there's a lot of ideas of biophysics underlying that. But from a pattern formation point of view, it's very interesting because you have a multipolar system. So you can get up to 40 fronts in such a system. So a lot of our work is thinking about just on the cell level, one cell in isolation, and how these patterns are able to form. What's also really nice in plants is we can isolate cells, make them protoplasts where they don't have any contacts, and we can also induce these type of patterns, right? And that's something that we're doing with collaborations in Tokyo, in other type of cells, xylem cells. But you get these, very classical Turing type patterns and so forth on the basis of the small g proteins. Okay, so that's one of the branches we're really interested in, the intracellular partitioning. However, in the tissue context, these cells, they have to interdigitate, they have to coordinate their cell shape changes with their neighbors. So you could think that this is just happening through physics, right? Because they're tessellated, they have to basically protrude and indent. But what we have also, we have mutants in which you can perturb certain biochemical signaling pathways that you're actually making them make holes. So they would actually make a lobe against a lobe of a neighbor and an indentation against the indentation. So you shouldn't take it for granted that these things are interdigitating, okay? So to understand this, so what's the process that these cells can communicate over the boundary? We actually focus some of the models and some of the work experimentally in looking at the interfaces and how the information of the individual cell polarity of every cell can be communicated. And I will already forward that in this communication, auxin plays a very important role. You guys have heard about auxin. The reason this was puzzling for us, that auxin could be involved in such a small-scale signaling event, is that auxins are quite quickly diffusible molecule, and we usually associate it with long-range morphogens or morphogens within a cell, as you probably heard. Now, a very important thing is that there are feedbacks between what's happening intracellularly and on the cell-cell communication both ways, and from the cell shape. So we really need to start untangling this, both through modeling and computational work, but also experimentally. And I will tell you how we're doing that experimentally towards the end of the talk. But what I would like to say is that cell shape, just the phenotype of being able to look at the cell shape, is very important for us because it tells us that, about many things, about what's happening intracellularly, but also what's happening with the communication process. So, a part of my talk will be about cell shape. Okay, we're going to deviate in that. But although that you might think this is sort of like a freak system, this pavement cell shape, with the understanding we're deriving from that, we're also applying it back to more classical, traditional tissues that you might be used to. So this is a plant tissue. This is actually the beginning of a leaf outgrowth. But usually when one thinks about auxin transport, and so we have these things here. This is a protein called a PIN. So it's an export facilitator of auxin. By the way, if anybody has any questions, just interrupt because, this is sort of some particular things. But what you see here is they're all aligned into a certain direction. So So this, you have this planar tissue polarity that you can see in the expression levels of this protein. And this is, for example, in Drosophila, so these are little hairs on the cells, and you see that they also have a certain alignment. And this is really, really typical in developing tissues, that you have a tissue polarity running across epithelium, right? Now, one of the things in all of these models is to try to understand, so how do these cells have this orientation of, proximal distal? And what we're arguing, and this will be clear by the end of the talk, is that we think to understand all of these processes, be it in animals or in plants, although they communicate in a different way, that the core element is to accept the fact that cells have the intrinsic potential to polarize spontaneously without there having to be an external cue which is polar. Okay? And we believe this very much from biological evidence, and also if we think about it evolutionarily. So we think that unicellular cells were able to do it, you evolve multicellularity, and so the tissue polarity that you see arising is using as a basic building block the fact that you can already break symmetry within a cell. So you don't need to have a neighboring cell to do that. Okay, it's a very harsh statement, but it also can be tested experimentally. One of the things that, if someone has a question, one of the things we also would like to address with this type of approach is this very fundamental sort of view on when people were thinking about, okay, like a file of cells, which were all the same, and they think, well, let's say we have a gradient spanning this tissue, right? How could one use the gradient information, the cells, how could they use that to be able to get an orientation? Now, one thing which is really important to think about is from the perspective of the cell, which is actually the way, biological information will, in the end, be processed. So one way is to say, well, these cells, they basically measure the scalar amount, so the average amount that, of that gradient that they are able to perceive. So that would be like here the figure C. So you would have this leftmost cell having more of that signal than the rightmost cell, and so forth. So basically you see that every cell, it only has a scalar. So to get some information of orientation, there has to be some type of comparison between neighbors, okay? The other thing that people can wonder is, well, maybe this gradient which is spanning over the tissue, a cell, it could be, for example, a ligand, and if the cell has receptors on the membrane, the fact that the cell already spans a certain space, that would allow the cell to be able to compare front and back concentrations. Okay, so that's another sort of generic way that you could imagine that a cell by itself could already get a gradient. This has issues, of course, because, for example, depending on the size of the cell, and also, if you just think about exponential gradient, these differences get quite shallow as you go to the tail, and so forth. In the end of the talk, I would like to show that by now supposing a third way of interpretation, that cells have an intrinsic cell polarity, we can, in a very robust way, start thinking about this in a totally different manner. Okay, so the polarity of the pavement cell. As you guys know, we could get polarity just with one molecular species being active and inactive. We don't need two. However, in this system, it is the case that you need two active processes, one active process telling you to become a lobe, and the other active process telling this region here to become an indentation. So you have two antagonistic pathways. So it's not a requirement for break of symmetry per se, but it is for the biological output here. There are two classes here. Basically, I'm just going to call one ROP2 and the other ROP6. But so the red guy, if you don't want to remember these names, it's just the front, and the green guy is the back, okay? Sometimes they refer to this as A and B. So if you're from the animal field, think about this as CDC42 and RAC, and think about this as Rho, okay? And so the green guy, ROP6, the indentation guy, what he's promoting is the bundling of microtubules, and this is sort of like serving as belts, holding the cell back from growth. And this red guy here, there's a lot of actin which is being recruited, again, through the very same conserved pathways, WASP, WAVE, and so forth. So we have actin branching, a lot of exocytosis, and the idea is that this is loosening the cell wall and promoting growth, okay? So these small G proteins, as you know, they can be active in the membrane, they can be activated through gaps, inactivated through gaps. They can be internalized through GDIs. This is quite important because then we have two populations, a fast diffusing population, a slowly diffusing population. And this is basically the switch, the core switch, which is really important to understanding all of these type of phenomena. In the particular case of this system, we have also a specific crosstalk between these small g proteins. So auxin is activating both, so which was quite counterintuitive at the time that this experimental data came out. ROP2 is inhibiting ROP6, and through an effector here, ROP6, we have an inhibition of ROP2. Okay, so we have a little thing here. So these RIC guys are effectors, right? So they're the ones which actually are doing things to the cytoskeleton as well. Okay, so we can just, you know, what does one do? You know, you start just rewriting that little figure in the most parsimonious equations, like ODEs that you can think about for ROP2, for ROP6. This is the active form of ROP2, of ROP6, and this is the effector RIC1. And auxin here is just a parameter which is like allowing this activation. Here we have the deactivation, so this is just basically the rate by which it's becoming into the inactive form. This model is totally conservative. We think that inactive plus active is always a constant. We're not considering production and decay. So we're simplifying it on that form. And here we have the mutual inhibition of RIC1 inhibiting ROP2 and ROP2 inhibiting ROP6. Okay, and we have another equation here for RIC1. So what we did is we put this term here in QSS to just be able to work with these two equations.
Quasi-steady state, is it?
Yeah, quasi-steady state approximation. It just makes things easier to, at least for me, to plot. So this is now auxin, which is this parameter, right? It's just this homogeneous amount of auxin. And what we see here, if everything is well mixed, we're not considering that the active and the inactive are diffusing at different rates. What we see is we just have like a little window of bistability here. So this would be the high amount of ROP2 corresponding to a lobe, and here to an indentation. I'm reviewing this very fast because this is something you guys have been discussing massively. Of course, this is just a standard stability analysis. The only thing it's telling you is that there is a certain regime of auxin which certain cells would be completely having ROP2 and some completely ROP6. It's not telling us anything about polarity. It's just about the total state that that cell would have. However, we would like to, in a quick way, probe for possibilities of the coexistence of the two states simultaneously in the cell, which is not the same as looking for the region where you have bistability. And for that, we use something that actually I started while I was in my PhD, which we call now the local perturbation analysis. And it's just a very, very nice, easy method of basically admitting the fact that the active form is slowly diffusing. So we can think about it spatially in the following way. So we have a global level of an active form, and we imagine what's going to happen if we perturb locally. So we have a local here variable which will be different than the global. And we ask it if we have a perturbation of arbitrary height, so it's narrow but of arbitrary amplitude, will this be able to reach a different equilibrium? Now what's happening with the inactive at that location, in this case we're totally conservative, the inactive of course would be depleted at that position, but because it's infinitely quickly diffusing, so that's the approximation we're at, it would flatten out. So we can basically say that for the inactive form, we always have the same amount, okay? So if we do this break of local to global, we can rewrite those two equations, but now we break it down in the local and the global form, right, for the active, which is the slow diffusing one. But of course, always where we have the inactive, we only have that one variable, which is the global, because that's always the same. And if we do that, what's really cool is that now, before we just had, right, here we had the stable equilibrium, unstable, and stable. So what we see is the global equilibrium, it became unstable at a certain region. And now we have these other branches, and they tell us what's happening with that local equilibrium. So basically, at a certain region of auxin here, we never get polarity. There's just one homogeneous global equilibrium. At a certain region here, you can effectively think that, well, there's a certain amount that you have to trigger locally, and then you'd be able to get a local equilibrium, so we'd be able to get the polarity. And then finally, after some more auxin here, this is actually going through a Turing bifurcation, but what we see here is that your global equilibrium becomes unstable, and you would always get spontaneous formation of lobes and indentation. So both of these equilibriums are possible. And these are just the simulations to show, right, this is a space and time that these are corresponding to these regions. So here we only have a rest state. We can never perturb that. If we perturb very slightly around this region here, we can have an insufficient perturbation that it goes back to a rest state, or a sufficiently big triggerable perturbation that we're able to get the polarization. And finally, over here, we always would get polarization. But what you can see is we can get, obviously, of different frequencies. Okay, but that's sort of in the Turing regime. Okay, let's go a little bit back to the experiments. So the lab of Zheng and Yang, what they showed, so NAA is a form of auxin. So that would be the parameter A, which was a bifurcation parameter. You know, if you give different amounts of auxin exogenally to the leaf, what you can see is you can get different numbers of lobes. And what was interesting in those experiments, they're sort of optimal. You know, so at a certain amount, you have optimal number of lobes, the most, and then if you give too much, it starts having less again. And actually, this sort of tendency we already see in such a very, very simple model, which is cool, but I'm not sure how meaningful it is. Now let's look at this spatially. So we take these equations, these PDEs, right? Because now we have the active form, the inactive form, these different diffusion. So what I'm just showing you is the active form here of ROP2. In red, it's high amounts. In blue, it's low amounts. And we are running exactly the same simulations on just a different, boundary condition, 2D boundary condition. Okay. Now, what you see in this system is this, the general behavior is that the interface between one species and the other, so this boundary here, over time it wants to minimize. Okay, this is now forwarding the simulation, so you can see that quickly. What also happens here is that these spots, they will disappear and they'll be incorporated to one region. And I think you guys have been discussing this in the last days. So your general tendency, right, in a circular domain would be just to have one front and one back. Okay, and here you have the curvature effect here of this boundary. Okay, it straightens out. However, those same equations run in this pavement cell shaped boundary condition. What you see is that you get trapped in these indentations, because actually to be able to overcome this, you have to go through quite strong curvature scenarios, which is very costly. So it's basically like a metastate. Yeah. So what we see here, the point that I'm showing you guys is that although biologically we know that these patches, downstream of them, they're going to cause things with a cytoskeleton, which will bring forth the shape. If you just give it a shape, you see that the shape also has an effect on stabilizing certain patterns. So there's another feedback arrow which is going through the fact that, you know, these things are diffusing and can't diffuse out of the boundary condition. So these are feedbacks that we think are very interesting, because as you start getting the pattern formation on the level of your small g proteins, on these diffusing proteins, and they will start altering the shape of your cell, it's very important to see how these dynamics are being coordinated, because they have feedbacks to one another. So we started looking at, we wanted to start to look at these dynamics, and we wanted to look at real leaves. So this is something that we initiated in the lab. Okay. And now we also started getting lots of interesting mutants, and then we had a problem. Okay, how do you quantify cell shape? And these are the type of cell shapes that we look at in the lab. So these are our leaves. This is a wild type, and this is a leaf I'm going to show you a lot. It's one. What do you guys know that leaves? You see these little mouths here? It has like these little funny cell shapes. These are stomata. You know, these cells, they open, guard cells. They open and close, and they allow the oxygen, the gas exchange of the leaf with environment. It's a little bit perturbing because they have like meristomoids and other things around them. So what we do is we have a mutant where we just don't have them, and then we can only focus on the pavement cell, which is our interest. Okay, so this is called speechless because they don't have the mouths. So sometimes I'm going to show you wild type and sometimes speechless. Okay, we grow them in conditions, by the way, where the leaf is still happy because we artificially provide the nutrients and the fluxes for this plant. This here, for example, is amnicerosa cells in the fly. So this is just to tell you, it's not only plant cells which do these funny things. You know, animal cells also have these multifaceted polarity structures. But anyway, how are you going to start classifying these shapes? You know, these are just like 4 shapes that we just chose. You know, they're different, we see they're different, we can talk about them, but how would we numerically quantify this? And so, I mean, we enter this field, and it's not like we want to invent something new. So what are the existing metrological instruments which people have for cell shape, right? Because it would be nice to use them and compare with other research which has already been done. So I will just quickly tell you what has been done, and maybe you guys can appreciate the problems we were having. So people in this field, what they do is they measure by hand the length of the lobes, and this is how they do that, and the width of the neck. So they go from one indentation to another. And they sum these numbers up and get an average, and then they score the cells. Now, what was really a problem for us is we don't really know what a lobe is or not. Would you consider, you know, this the lobe, or that the lobe, or is that little indentation already a lobe? So it's very arbitrary for you to decide what is a lobe, what's an indentation. Also, what is a neck width? Is it from here to here, from here to here? How do you choose what a neck width is? The other thing that you see is that these cells, they grow a lot, right? So this is just like a length. It's very different to compare shapes of different, yes?
So when you're talking about shape is something that's scale invariant, so it's not something that depends on, quote, metric size.
I totally agree. I totally agree.
So
yeah, so we're going to go to that. But this is just what people had at the moment. So, you know, these are papers like Nature and Science, which are saying this is how we classify the pavement cell phenotype and shape.
But in medical science, the word morphometrics is used sometimes.
Yeah, and one of the things with medical science which is really, I mean, there are lots of shape descriptors for cells, but where you need a landmark, you need something to say this is the beginning of the lamellipodia and so forth. So all of that we couldn't use because there is no landmark here. There is no start and end of that cell that you can be able to distinguish. So anything where you need to say, oh, this is a tip or something like that, we can. So all of those type of morphometrics, you know, that people use for fish and for skeletons, you know, throw away. Okay. So basically, you know, they use these average lobe lengths and neck widths. But what I can show you here already, I mean, this is a wild type cell. These are mutants, RIC1 mutants. You see that they have really different cell shapes. And what you see, it sort of blurs out with these type of statistics, you know. They almost get like no differences. So you see, it's just, it's quite lousy. The other thing that people use very much is what they call the circularity. So they say, okay, let's just take the area divided by the perimeter squared, and then we get sort of how much deviating you are from a circle. And so you get these sort of scalar fields here. But you know, if you zoom in and you can start appreciating like zones where you have the same colors, and actually cells are very different there. So you can still also have quite a lot of generalization on that level. Another method that people use is called skeletonization. This is sort of a computational method, so it's an algorithm. So you take your pixelated, your voxels, your image, and you just strip it down through an algorithm, and then what you get back is just a skeleton. And then you count the endpoints. Now, we tried this out, these skeletonization techniques, and the thing is that you have some parameters there, exactly how you strip down these pixels. And it's very sensitive to how you tune that, so you'll get different number of branches. So again, this is something which is very much sensitive to parameters, but also an issue we have is that it's very much dependent on the resolution of your control co-image. And it's also very then dependent how big your cell is in relation to that resolution that you're using. So, but this is, you know, these are people which are using it for something which is like the same resolution, and then they count these number of skeletons. So the ends of that, and so this is, for example, wild type, and this is, by the way, this is a GEF mutant. So, again, people realizing these issues, they try to combine them, so you say, okay, we sum up the skeleton ends plus the circularity, and we get an average score. And I mean, I can go through all of this, but you really start seeing contradictions in the way that these things work out. And if we apply that, just like a proof of principle, to those four very simple cell shapes that I showed you in the beginning, for every different metric that you would use here, like these two would be the ones which are most similar to each other, the magenta. These two here would be very similar to each other if we would look at the average width of the necks. If we look at the form factor, for example, these two would be identical and these two would be similar. The aspect ratio, suddenly the last two would be basically identical. The skeleton endpoints, the first and the third column would be identical. You know, it's just all over the place. And then you could also say, well, why don't we just use a combination of all of these metrics, right, together, and then you could do some PCA. We could do that, but what we realized, we didn't really get any understanding of what that meant for the cell shape. And so the point I want to make is that any type of, like you take a globe, which is in 3 dimensions, you try to collapse it onto something, project it onto like a lower dimensionality. I mean, you're always going to be losing information, right? So it's not that I'm saying that I want to, have the full information, but I would like to choose for myself a projection that I find meaningful for the type of problems and questions that we're addressing. And it's actually quite interesting. I mean, you have these different techniques of projecting the globe. And I saw this cartoon, I just had to talk about it. So it's about saying, like, what is your favorite projection, right? Your map projection. And I recognize what my favorite one was, which is this one. So I really like the hobo dia. And because I come from Brazil, and this one is really nice because the equator is, it's not squished downwards. And so here it says, okay, what it tells about you. You want to avoid cultural imperialism, but you've heard bad things about Gal Peters. You're conflict averse and bioorganic, and I was like starting to feel embarrassed here, blah, blah, blah. Okay, but what I want to say is a method I'm going to promote here. I'm not saying that this is the method, this is the best method, but this is the method that for us and for our type of, where we're coming from, was was the best. And where we're coming from, so this is something very personal, so I really like music. So the inspiration I will give now is really thinking about how do we identify a note, okay? So if you play a on a piano or A on a violin, right? You can just take the Fourier spectrum of that, and you can say this is the principal component, the principal mode, and that frequency will be A. Okay, so that is possible. I mean, we can quantify that. We can quantify its sound, and we can characterize A from a B flat, and so forth. So we would like to have something very similar to that. However, we would also like to have something where we can distinguish what is like A played by a clarinet and a A played by a violin. Okay, so the first mode would be the same, but we want to keep the whole spectrum and look at the timbre. So look at all the other components which are there and take that into account as well. So I will show you then the problems that we're dealing with. This is a wild type leaf. This is the pin 1. This is the one which is having an issue in the export of auxin. And it has defective phenotype because there's something happening with the way the cells communicate with one another. And we want to quantify these type of things. So we want to make this biological interpretable. This is another thing that we're working in the lab. So this is all a picture from the lab. This is the wild type 10 days. And at 10 days, I mean, you can see the shocking difference here of the shape. This is a leaf which is not being able to produce enough auxin. So here you see really that the intracellular polarity is being affected. But we really want to be able to take all of these cells and then make it statistically amenable. So if I would just give you a cell, is it closer to a wild type or is it closer to, for example, one of these mutants? So we really want to work with large populations as well. So because we want to work with large numbers, and we also actually want to look at the evolution. So this is a leaf at 7 days, and this is at 23 days. We actually have all the time courses in between. So I can tell you more about this later, how we do this. But what we would also like to do is we want to start like tracking these cells. And what we realize is that we can't do that by hand, right? We can't segment and do all of these things by hand, so we really need to look at automatic approaches and automatic approaches for the shape analysis. Second. I thought I had a movie here. Sorry. Maybe here? Yeah. Okay, so what we use for this, so this is this is the confocal image, and this is how we would get our segmentation. I'm not going to go into the details, but we use, if you guys ever heard about the Cellar-Potts model, which I think you have, we use the Cellar-Potts model to segment cells in a confocal image. We call it the segmentation Potts model. Okay, but it's a great way to work with noisy data and to get your cells back. And once we get the cells back, we would be able, for example, over time, also to start like comparing, what's the differences between cells here which have not yet undergone the multiple cell polarity, and to those cells which already are a little bit more mature and have already suffered these cell polarity changes. Another thing I would like you to appreciate in this picture, the scales are very different. So these cells can, in afros, they can be like 50 times bigger than the cells down here. Okay. So we really also have to have a method which is not going to be size dependent, which is just like on the shape level. So these are the distributions that we end up with when we do the segmentation. So we see like this huge variety of cells. And yet we think that there's something which is very characteristic about these cell shapes when they have matured. So we end up with all this type of data. How do we approach it?
Are those the notes?
These are the cell outlines.
I'm saying, are these the notes that you're going to have?
Yeah, these are the notes. Yeah, so I want to listen to it. I want to start listening to this and see what are they playing, right? Also notice that these are non-holomorphic shapes. So if I want to now start describing the outline, I can't just do that by saying, okay, I take a point from the center of mass and just look at the radius and the angle, because I wouldn't get a function for that. Like, for example, here, for one radii, I would get different points, different intersections. So what we do is we do a Fourier analysis. That's how we begin with. That's the first thing you think, right? But basically where we have to use the x and y position for every point, right? So this is what I mean by non-holomorphic, right? So if we would just do something like that, we'd have like different points back. But so we take the x and y component, and then we're just going to describe it as the sum, which will go up to n, which is basically, in this case, given by the number of points that we have, so the resolution of our confocal image, and then we sum the progressive modes. Okay, this is the elliptic Fourier analysis because we're doing it for x and y, so we have these matrices. So we parameterize it, so when you do the complete full circle, you have this, you complete T, big T. Okay, how exactly, I mean, it's obvious, we all know how our functions work, but how exactly do you add up matrices, ellipses, and get back the shape? Okay, now it is very important for this talk just to think about this, how this happens, and it's really like this game, the spider graph. So, you know, you have this one circle, and you have a circle which is rotating, you put your pen in, and then you just like start circulating, and it leaves a path behind. So I will show you how we get from a shape, you know, we decompose it in these matrices, and then as we sum the first rotor, the second rotor, the third rotor, how we start getting back the shape of the cell. Second. Okay, so this is the first ellipse, first mode. This is the second rotor. What happens is you have an ellipse which is rotating once around, and you have a dot which is rotating twice around. So this is the dot rotating twice around. It leaves a green outline. Now you put a third ellipse which rotates 3 times around the second rotor. Then you'll have a fourth rotor which rotates 4 times around the third rotor, which rotates 3 times around the second rotor, which rounds twice around the first rotor. Okay? And so basically that's the way that it goes a little bit quickly. So this Mexican hat is your pen, right? And the rotors will be these will be the harmonics. So this is mode 1, mode 2, mode 3. And this is how you're starting to get back the description of your cell. Okay? So what's really important here is the ellipse, the angle of the ellipse, the starting point of your pen. Okay, that it gives back the shape, that's per construction. That's not surprising. But it has an issue. Okay, elliptic Fourier analysis for shape has a huge issue. And that's why you see many publications starting to use it, and then people abandon it, and if they do PCA, they get no results which are consistent. And the reason is that, first of all, it's totally degenerate. There are many, many combinations and positions and starting angles which give you that same shape. So looking at those numbers actually doesn't really tell you if one shape is closer to another or so forth. The other problem is you don't have your note back, right? So for example, if I would just now give it a very simple shape, 1, 2, 3, 4, 5, 6 lobes here, you don't have the mode on 6. Because of the way that these things rotate around each other, you have relative rotation effects. So you get your peaks at the mode n plus 1 or n minus 1. So you see a peak at 7 and 5. You don't see it at 6, which is actually what we would want to have, because we would like to have a phenotypic correspondence between these numbers and the lobe numbers that we're seeing. I mean, if something's not clear, just let me know. So this is, for example, just illustrating that. So what I did here is I added a third rotor to the first two, which are exactly the same. And just by changing the third rotor slightly, what you can see is you can either get 2 lobes or 4 lobes or something in between. Okay, so adding the third rotor is not saying that we're getting 3 lobes or something like that. So this is something we would like to change. So the elliptic Fourier analysis is degenerate. We can see this in this case. And the biological problem is that we don't have this trustworthy correspondence between the FAs and the actual phenotype. So what we did, we just moved to a different basis which uniquely represents that shape, okay, and something where we also have a perfect match to what we would call a lobe, okay. And it's very simple matrix operations that we do, but basically we take the core matrix which is representing these rotors. Well, first of all, we have to make sure that we have to always start with the same orientation, counterclockwise orientation, just per convention. We make sure starting points are always reset, so we don't have degeneracy. And then under that new matrix transforms, we actually then split the so these rotors up into 2, a symmetric and an anti-symmetric component. And then we sum them up together in a new basis, which we call the local FA. And this is basically what I'm doing. So I'm saying that for the elliptic Fourier analysis, we have those matrices which give those ellipses, and now we break them up into 2 circles, clockwise and the anti-clockwise circulating circle. Okay, so the plus and the minus. And now, so this is for mode n, so we'll have a lambda plus n and a lambda minus n. And now we have to combine them to give us these new harmonics, which is going to form our new basis, which we call the local FA, which is the lobe contribution FA. And that's done by taking the n plus 1 component of the plus and the n minus 1 component of the minus, and we sum them up. Okay. And so what we do with all of this is basically, this was our previous scenario where we had the peak on 5 and 7, and now we nicely get the peak on 6. So it works very nicely. So let's see, are we in tune? Can we really now listen, for example? So what's happening if we reconstruct such a trivial shape with 6 sides? This is the shape that we get if we just truncate the series at 1, 2, 3, 4, 5 using the elliptic Fourier analysis. And what you see here is already at 5 you were getting some type of shape, at 6 you had the shape, and at 7 you had it perfectly for the elliptic Fourier analysis. Well, what we get now for the local FA is that we continue more or less with a circle, and we only recuperate the shape which has its principal description at 6 at 6. So this is exactly what we want to have. Saying it in other terms, if we were just like give it like purely geometrical shapes, we always see the peak. So what's the major note that's being played? So here we see it's 3, here we see it's 4, 5, 6, 7, and so forth. So we really are able to count these sides or these lobes. Okay, another way now we can start looking at these things is, again, so this is our original shape. This is how we would do it by reconstruction through the local FA. And now we can ask the question, okay, if I would truncate it at 1 and I look at the difference, so how well is just the first harmonic able to describe the shape? So we take the difference between this shape and that shape, the area difference. We see it's very bad, right? So we have a big difference, discrepancy. But we see here, for example, at 8 it's getting pretty good, right? So this has 1, 2, 3, 4, 5, 6, 7, 8, a little bit like an 8, 12, right? So you can start seeing how do the modes contribute to explaining the shape, and that's something we call the XOR difference. And so if we again just run this on the trivial shapes, we see that indeed here, like we had a big discrepancy in the shape, and then suddenly it was resolved at 3 because it's a triangle. Here, in this case, it was resolved at 4 because it's a square. Here it was resolved at 5, right, because it's a pentagon and so forth. And if we take basically the derivative of this, so what is the mode which has best contributed to the explanation, we get these very nice peaks. So this is what we call the marginal difference. I mean, they're just ways of doing that. But what's really nice with this now is that not only can we quantify, okay, this has 5 lobes, 6 lobes, 7 lobes, we can also see the degree of amplitude. So we can see how loud that note is being played. So this is like 6, 6, 6, 6. And then we want to talk about timbre, right? Those pavement cells, they don't have these perfectly aligned symmetrical shapes. You know, they come in all of these different flavors. So these are like shapes where they all have 6. You would count 6. Here you just see like a very simple peak. Here you start seeing that it has something like a 2, it also has something like a 4 because it's a bit squarish, so you have these other modes, but again, you always have the peak on 6, right? And this one has the lobe of 6, but it also has different components. So basically, this analysis is saying it's very elongated, it has a very big thing on 2, it's not symmetric, it also has a type of triangular feature, so it also has the 3, and it also clearly has the 6. So this is a non-discriminant way of us starting to quantify the lobes. And if we look at the whole spectrum, we can also classify, for example, the entropy. So, you know, how complex are these shapes? Okay, so I'm going to go a little bit faster because of time. So when we do our cell tracking now, we can start looking at how different modes are coming up over time. So here we had something where we see 5 and 7 is coming up. We have very sometimes simple cells where they just like elongate as a rectangle. So basically, by looking at these things over time, we can really see the progression of the cell shape. Because it is a basis which is uniquely describing the cells, we also have the landscape. So this is the modes over time. And we can start seeing when do new peaks form, right? So automatically, we know the times where we're getting new polarity outgrowths. And so we're using this method to go back into our massive data set to be able to find, for example, like this lobe is splitting in 2, and this lobe here, it is trifurcating in 3. By the way, this is not the only way that lobes appear. Sometimes they just appear equidistantly between others. Okay, so it's also interesting from a pattern formation perspective. Okay, so what about now tuning into the populations here? I'm just talking about one cell. So we want to listen to all of these notes at the same time and try to get something reasonable. To be able to validate our system, what we use is the Seller-Potts model. So as you know, the Seller-Potts model in its most basic form, it just works with cell adhesions, right? And because it's cell surface tension driven, basically what you have is you have like rounding up of cells, or you get them in these type of packing configurations. The anisotropic. What we do there is then we start adding certain internal polarities to be able to skew these forces, so we're able to basically manipulate for parameters. We're able to, for example, instead of having the resting shape, equilibrium shape being a circle, we can get it being something like rod-shaped, or for example, we can give it multiple polarities. So in this case, you know, we're imitating like a pavement cell. It's a very artificial system, you know, because we're sort of like pre-describing what the specified shape is of your Seller-Potts model cells, okay, through the energy, through the Hamiltonian. But what's really nice is now when we bring these cells together, because they're packed, they cannot just realize that specified shape. They'll have to reorientate themselves, and sometimes they won't be able to make those desired lobe numbers. So what we're showing here on top is the Seller-Potts model if it were in isolation. So this is the shape it would want to realize, and these are the resultant shapes it ends up getting when it's in confluence, when it's in the tissue. And so what we see here, the colors, so this is the lobe contribution 4, 5, 6. So obviously 6 here is very prominent, it's very red, because this is the major harmonic, which describes that shape. But we see that L5 is actually happening in the tissue context more than you would expect from just the isolated cell. L4 is also happening more than you would expect in the isolated form, and so forth. So we see the distributions of shape that you get due to the conflict of having to resolve your specified shape within a tissue conflict. But the reason I'm showing you that is we can do statistics on this. So we can take averages, means, and we get very interesting results about how your desired shape is changing due to the insertion of the tissue it's in. Okay, I think I won't dwell too much on this, but we're using that for the in silico tissues, which are up here. And we're also using this, actually this is not in silico, I'm sorry. This is real tissue, okay. So this is real, this is the Arabidopsis wild type, and this is the Arabidopsis speechless. So we're actually now, when we quantify, okay, so which cells have most 4, 5, 6, 7, we see very similar distributions. The eye-opener for us and for biologists is it doesn't mean that this cell had wanted to have 5 lobes. It's a result of some type of polarity which was internal to it, and the fact that it has to resolve that shape conformation within the context of the leaf. So let's do some statistical analysis. If we compare wild type with a GEF mutant with a RIC1 overexpression, okay, these are things for all of which we have simulations for, and we understand how this would perturb the cell shape. if we would just look at what people would call the main anisotropy, which is basically the first mode, so, or the inertia matrix of the shape, you see that they don't seem to be very much affected. And so people could conclude that these polarity factors don't do very much with the polarity because the cell is just, overall, is just as much elongated. But it does massive difference by the complexity of the cell shape. So basically, this entropy of that distribution. So here we see very simple shapes, so we don't have all those lobes, and we have massive amount of lobes for the wild type. So suddenly we can quantify these things very precisely. Okay, so on a real leaf, as I was telling you, complexity starts getting increasingly more complex at the tip. And what we see is that over time, if we just accompany one cell, its profiles of these modes is just sort of continuously changing. So of course it becomes very different in the end, but if you take one time point to another, we have a smooth transition. So we thought this is quite interesting because it can allow us to do something very practical, which is cell tracking. So we have segmented already these cells, which I told you. So we have that. We have all the segmented cells. But what we don't have is we don't have the match of one cell in one time frame and that cell in the next time frame. And the way that usually has to happen is that people have to say this cell is that cell, this cell is that cell, because we lose XY information. These leaves are moving and so forth. So what we can do now is the computer, it just knows what the sort of the fingerprint is of the cell through the L numbers. And the other time frame, it can say, well, this is the most probable one because it has the most similar fingerprints. These are the other ones which have similar fingerprints, but they're off. So it knows this cell is that cell. So that's what we do here. So basically, it just like says, okay, these are the cells, the first 4 cells which I know how they are. And it starts taking a little bit of the strain field around it, and it starts iterating through the L shapes. And through some iterations, we get a perfect tracking, right, between one time frame and the other time frame, but using now something we invented just to classify cell shapes. Okay, so basically, if we start looking where these cells were, where they've gone to, we take, this is the velocity, we take the derivative, we take the strain in relation to the center. What we also get for free is we get the growth rates at different regions of the tissue. And we can then count divisions, because what the algorithm also does, we can say, okay, these 2 cells here, if we add them together, it gives perfectly that cell shape back. So it can now quantify where the divisions have happened. In the quantify, it can report that. And what we have now, we have basically for all the cells over 15 days, we have its progression and its time dynamics. Going back to the beginning of the talk, this is what we think is really important because we want to study the combination of the intracellular time dynamics of those patches together with how the boundary condition is actually changing. So now we actually have this information. But we also have information of, for example, how long the cycles are between divisions, how they're happening for different lineages. And we can also quantify that automatically, right? So this is basically how cells are moving along the leaf. This is time, this is the distance from the base of the leaf. And this is the algorithm quantifying where the divisions are happening. So what we see is at the base of the leaf, we have a meristematic zone. Then when the cells leave the meristematic zone, they start growing. And that's also when actually this whole polarization business enters phase. I won't go too much about that. But so we're integrating that information on the scale of the leaf. And I see I will just use another 5 minutes to finish this story about, so that was a huge detour about what we're doing with the experimental side to be able to capture a little bit of these dynamics. What about the way these cells communicate to one another? So this is a really bad picture, but at the lobes you also have the expression of pins, okay? So there are more pins, which are these exporters of auxin. Now, until now we were regarding auxin just as a homogeneous parameter, which was activating both pathways of lobe and indentation, but now we see that where you get the lobe, you get more pins. So could this be helping to coordinate the interdigitation of these cells? Now, I forgot to take this away. Okay, I forgot to delete it. So it so happens that the community, we know that there are receptors at the membrane. We know that these receptors are sensitive to auxin. It was thought until a couple of months ago that this receptor would be ABP1, but then it turned out that that was a huge genetic mistake. ABP1 has nothing to do with the story. But still we know that there is a membrane response, but this here was really, really bad genetics, really, really bad biology, so ABP1 has nothing to do with it. And this is getting me into some problems. So we don't know what the receptor is, what the protein is. We know there's something there, listening to auxin in the cell wall, okay? We also know there's a canonical pathway, but basically what I'm telling you is that if you add auxin, what we can see in protoplasm, for example, is that the active ROP2, it starts activating in minutes. So this is not going through transcription translation. There's a receptor at the membrane which is able to activate gas. Receptor of mystery X. We don't know what it is. Okay. So the problem is, how can we get, if we have a very small interface, which is the cell wall, right, because these cells are not able to touch, ROP6 is not able to touch ROP2 from the neighboring cell because there's this cell wall in between, but it is able to export auxin in this cell wall. The question is, would this be sufficient to cause the opposite response between one interface? So one cell has ROP2 and the other cell has to have ROP6 at that place, right? Because that's how indentation and lobe and indentation meet. So we call this the Battenberg paradox, right? So you have a thin jelly and you have to communicate that information and get the opposite response. La la. Okay. The first thing that people thought is you could just do that with a positive feedback loop. So basically, if you have auxin, you promote ROP2, ROP2 brings pins to the membrane, pins export auxin, and there you go. Okay, so what you can show is that if you have just that, so we forget about the intracellular polarity, we just say everything would be through that amplification of a certain auxin signal, we don't get anything. So this is auxin in the cell wall, this is ROP2 and ROP6 in cell 1 at the membrane and on the other side. So this is just a sandwich between looking at two interfaces. The reason this is not working is because auxin is fast diffusing laterally, so you're not able to maintain, right, and amplify this positive feedback loop. You can really mess around as much as you want with those parameters, but it just doesn't work. So we thought, okay, let's put in the intracellular partitioning mechanism. So we have both of these so ROP2 and ROP6 in both cells, so yeah, so they will break symmetry, we know they will. But now everywhere where you have ROP2, you get exportation of auxin. But what we see actually is on the other side of the membrane, ROP2 also accumulates, right? So basically we get a contradigitating pattern. I mean, it's a perfect communication, but it's totally the wrong communication that we want to have. So we have the same response on the opposite side of the cell, right? So we have very high auxin here in the middle, or higher auxin in the middle, and ROP2, ROP2. Okay, so we have this. So we look back in these profiles of how ROP2 and ROP6 were being activated by auxin, and we realized that although both of them get activated, the ROP2 activation saturates more rapidly than ROP6. So ROP6 is basically getting, so this pathway here, ROP6, it's asymmetric. It gets a little bit more advantage from auxin than ROP2. So this is just another way that we like to represent it, just giving A and B, so people don't have to, remember all this nomenclature. So basically, we have one of the components, which is the lobe, which promotes the export of auxin through pins, and that pin is promoting the activation of the antagonistic pathway, which is the B star, which is the activation of B, right, of ROP6. So if we promote this asymmetry, let me just show it to you here first. Starting from an initial random condition, actually initial rest state, what we see is, okay, we get the break of symmetry, and suddenly, you start seeing it happening here. You start seeing that you get this really very nice and very coordinated interdigitation. So where you have ROP2, on the other side, so you have auxin, ROP6 wants to localize itself, because actually it's quite intuitive, ROP6 is getting more advantage of auxin. And then ROP2 on the other side will locate to the side. And here you get ROP6. So you get a perfect interdigitation. So basically you get this bifurcation in the type of patterning you can get. Either you only get contradigitation, you make a parameter change, and then you only get interdigitation. Okay, and it's very robust. And it's so robust that even if I would start it in the contradigitation phase, which you could think could be stable, it unstabilizes any noise and goes into the interdigitation. Okay. So what we want to do now is, and that's actually we are doing this in the lab, is we want to have now, we have those total mutants that I showed you. The problem is the whole leaf is affected. So what we're doing now is we use a system, a Krelok's recombinase system, where we can, at a certain given point in time, we can cause a mutation of an overexpression of either the small g proteins, either its effectors, which are very much involved in the way these small g proteins crosstalk, or, for example, the activators, so the GEF and the GDI. The GDI is really important because it will change the diffusion. And then we can, for example, make a wild-type mutant, a mosaic. So we have one cell which will become different. It's going to have daughter cells which are different, and we also have a color for that. So we know which cells are affected. But the nice thing is, it's in a sea of wild-type. So what we can start doing is we can compare now what is the difference in the polarity, the number of lobes, the way it's indentating, right? And we can compare that to the interface, which is really important, the interface of a mutant cell and a wild-type cell. So the questions we can address is, if you are a cell which has the correct polarity, are you able to induce the correct polarity in a neighboring cell which does not have it at different levels? The other thing we can do is we can then say, okay, all cells have the correct intracellular polarity. The only thing we're messing around is the second part of the story, is the way they communicate that polarity through the pins, or, for example, how they localize the pins. So that's also very interesting. So now we know that the intracellular polarity mechanism is working well, but just the communication is not. So then we can also compare now what's happening between the two mutant cells and a mutant and wild-type cell. And this is the only way that we could devise experimentally to start isolating these different effects of the interaction between the cells and the intracellular pathway. And it will also give us a lot of ideas about mechanics, because as you know, until now I haven't been talking about mechanics. I'm sort of trying to leave it out, but we could see what the effect of mechanics would be on that bit. And by that I would actually like to cut the talk short, but just say that although we're really interested on these facets here that I've been telling you about, these models we can generalize not just for interdigitation, but also for tissue polarity. So why cells would align over larger time scales, larger space scales. And that's actually, the core mechanism is fundamentally the same. That you have auxin, which is a signal which is trying to minimize itself in interfaces, and that allows alignment of cell polarities, either over interfaces or over whole cells. So, this is the last slide, okay? And I would like to thank for this part of the talk Samaray, Yara, which did a lot of the imaging work, and these are the students, the postdocs, which at the moment looking at the leaf analysis. So how is the meristematic zone of the leaf being regulated? Susan Duncan, she's designing the Golden Gate mosaics in my lab, and with Benish we're actually interfering in the cell cycle during the leaf growth to see how things are working out. So thanks a lot for your time.
We've probably got just enough time for one quick question while the other speaker gets set up. Of course, there'll be lots of time over coffee as well. So anybody pressing question for Veronica?
If you take the curvature around a given cell, that scales like 1 over the length, so you can non-dimensionalize it by taking that relative to the length. So for each cell, you would have
the relative curvature of one lobe, right?
No, the curvature distribution around the entire cell.
Yeah.
That scales like, the curvature scales like the length, 1 over the length. So we scale it relative to the length of the cell.
Or then you get one value which is just telling you the total amount of
distribution of the curvature across the whole
yeah.
family. Have you looked at that over the entire population?
Actually, it would be nice to compare that, because I think that would be a really nice metric as well. Yeah. I should have a look. What we like about this other method, I would say, is that it's very cost efficient, and it can also give you the numbers. So we are sort of interested, like when you go from a discrete number 5 to 6 to 7. But I agree. I think your method would be very good. Yeah. Should we do this one? Do you need this? You can use mine. Oh, you have it. Sorry.